So yeah, I will have to open that because just give me a minute because okay, so let me know because at what point do you want me to stop?
Yes, ma'am, from 14th.
From slide number 14th. Okay. All right. So what is your question in this? So what is the question about this slide?
Yes, ma'am. No, ma'am. The last part where the reaction was taking place from where I mean, there was one product which was forming, which was excreted by the kidney. From there, I guess. I don't know the reaction. I forgot.
I see. Um, so anyway, I think let me just repeat for the benefit that I will just go back very quickly to slide number 13, where through genome mapping, because you see the investigators were in a position after the protein was purified, you can always load this purified protein on an STS-based gel. And since the protein is already stained by Comasi blue, you are also able to track this particular band on the gel. Okay. This band can be extracted out from the gel. It can be subjected to hydrolysis by trypsin. So trypsin is a proteolytic enzyme which only cleaves, for example, the peptide bond, provided the amino acid which is on the left side of the peptide bond is going to be either lysine or arginine. So that way, when you do trypsin digestion, there are very defined protein fragments which are going to be generated, okay, out of the larger protein. So therefore, every protein is going to have a trypsin fingerprint because lysine and arginines are going to be located at very, very defined locations in that protein. So on the basis of that, the protein is extracted, a fingerprint is derived, and some of these peptide sequences were now they can be translated into the DNA sequence, or they were mapped to see whether these proteins are found elsewhere. So in this isolate, for example, okay, through peptide searching, etc., it was found that there were two new genes which were located here, which were called as ephedrine dehydrogenase and pseudoephedrine dehydrogenase. Okay, so this part, you should be able to correlate as to how, beginning with a protein which you have purified, then you are able to get what is the corresponding amino acid sequence, the peptide fingerprints of that protein, and this can be then mapped back to the genome to see if there is any DNA sequence which is going to code for peptides, which will have a very defined sequence. In this way, on the genome, the gene corresponding to that protein can be located. Okay, so once this gene you have already mapped on the genomic DNA, and the entire genomic DNA can be extracted, primers can be raised. So a forward primer here and a reverse primer for ephedrine dehydrogenase. Similarly, forward primer here and the reverse primer here. And if you start out with genomic DNA as a template, you should be able to amplify out both the gene which is coding for this protein and the gene coding for this protein. Okay, and eventually they can be cloned. So that's what we have here. So now in this slide, okay, what is therefore put down? Because remember that in this particular bacterium, when the bacterium was incubated with only ephedrine as a carbon source in the material, or this was the first product which was detected in the culture supernatant. Okay, so do you understand that? Because, methcathinone, if you will recall and go back to that, earlier slide, that let me take you back to this slide number 8. Okay, So the data which was obtained from this bacterium indicated, like the triangles here in the figure, they refer to the concentration of methcathinone. Okay, and in the triangles they represent actually that this particular product is also formed. Okay, so what we are trying to say, in other words, is that these enzymes which you have been able to identify, ephedrine dehydrogenase and pseudoephedrine dehydrogenase, are capable of converting these particular substrates into methcathinone. All right. So
Understood.
is that understood? Why they are trying to bring it down here is because this is the stable product which you have detected in the culture supernatant. Then in the second slide, what has been put down is that how is it likely to be formed? There is an explanation as to how methcathinone, as well as so many other products which were detected in the culture supernatant by way of GCMS, how they could have arisen. Okay, the group is basically a chemistry backed group. So they were based on the known chemistry of the dehydrogenases, etc. They have put down this particular pathway that, starting with pseudoephedrine, if you have a dehydrogenase which is acting on it, which means removal of hydrogen. Okay, so hydrogen is taken away from here, and this is how the methcathinone is going to be formed, because the action of this dehydrogenase is going to be able to remove this hydrogen, and that is how this product is formed, which could be detected in the culture supernatant. Such a product, however, is highly unstable, and it loses in an aqueous solution, it loses the amino group. So you have the formation of phenyl propane 1,2-dione, which you also detected in the culture supernatant. And then again, by the action of dehydrogenases, you have the following reaction which is occurring: phenyl acetyl carbinol. Okay, so so there is an addition of a hydrogen here. Nevertheless, dehydrogenases are capable of doing those kind of reactions. Either they can add, however, it is NADH dependent. So hydrogen is being added. Here it is NAD dependent enzyme. So you have NADH as the product, but here it is NADH as the substrate. Okay, and then it can be converted into benzaldehyde and finally into benzoic acid. Now, benzoic acid is a metabolite which many of these bacteria, soil bacteria, are able to degrade down. It is an aromatic amino acid which is readily attacked by many enzymes which are already present in the soil bacterium. Okay, and so what it means is that there is a complete mineralization. Benzoic acid eventually will be converted into carbon dioxide and water by several enzymes, just as glucose or lactic acid or any other carbon compound can be metabolized. Same way, this is only a C and an H compound. So it is completely metabolized to carbon dioxide and water. That is how it can serve as a source of growth. Or in other words, going back, okay, so this series of events which are occurring allows ephedrine to be used as a sole source of carbon. And nitrogen, as I explained earlier, pay attention to that this nitrogen, because nitrogen had to be added from outside, otherwise Arthrobacter was not capable of growing. That means the nitrogen which is contained in ephedrine or in pseudoephedrine, okay, could not be used as a nitrogen source. So this NH2 anyway leaves the system. So the cells therefore are dependent on an external source of nitrogen. So is that clear now?
Yes, ma'am, clear.
Okay. Anybody else in the class? I mean, do you have any questions regarding this? Or any one of these other slides which we have in this lecture? So quickly, yes or no, students, so that we can move forward.
No, ma'am.
Okay. So please, read that paper, because I don't think that you will be able to understand.
Yes, ma'am. We'll read research paper.
Read them, because it's a very nicely written paper, not very complicated to understand. So now let me go with the biosensor PPT. Anytime if you feel, because my this thing is showing that there is a little bit of a problem with bad network. So if you can't hear me, or if the slides are not moving, then let me know, please. So somebody will have to give a cue that the slides are also changing. So can you see the slide number 6?
Yes, ma'am. Slides are changing.
Okay, so you will remember last time that on that slide, if you had a chance to read, that there were several, sensors which were described, that is the biological sensors, which are based either on enzymes or antibodies, etc. Or we also, I had put down, to show you that how, if microbes are being used as sensors, how these are likely to be advantageous, because these are very inexpensive and microbes can carry out series of reactions. So therefore they can be very versatile, unlike enzymes in which only one type of an analyte can be analyzed, or antibodies are very specific for an antigen. So if you use antibody to detect something as a biosensor, then it becomes highly specific. Okay, so here is an example that just to give you that if, for example, urea is to be detected, because you will recall that whenever you go for a blood test, okay, glucose is always measured, plus urea is always, the doctor will tell you during a blood test that get your urea also measured in the blood. Okay, so just like glucose sensors are important, the urea biosensors are also very important. And these are based on using urease as an enzyme. Okay, so glucose, for example, I had given you an example that glucose can be monitored by incorporating glucose oxidase. So what you see here, for example, is a urea, and you want to detect what is the concentration of urea. So for example, a drop of blood is put, and then what is going to be detecting it is the enzyme urease, which is given in black here. So urea is broken down into carbamic acid by the action of urease, okay, on the surface of the transducer here, for example. Okay, so let me know if you can see the slide.
Yes, ma'am. Slide 6 can be seen.
Okay, all right. So I have not changed the slide so far. We are still on this slide number 6. All right. So you have formation of carbamic acid, and it undergoes an automatic reaction on the surface of the transducer, so that carbon dioxide, nitrogen, and protons are released. Now, under amperometric detection, okay, you have the electrode potential is maintained constant, and the flow of electrons which comes as a consequence of this reaction, okay, is going to be measured in the form of a current. So that is your amperometric detection, and the current that is produced is directly proportional to the concentration of the urea. So that is how you are able to quantitate it also. So you are able to measure that urea is present, yes, and how much is by the extent of current which is going to be produced through amperometric detection. Okay, under potentiometric detection, for example, what is done here is that on the surface of the transducer, okay, there is a different change which is occurring, where carbamic acid, for example, releases ammonia and bicarbonate ions. So this reaction causes a change in the pH. Okay, or in some other potentiometric sensors, you could detect a change in the sodium or in the potassium concentration, etc. So accordingly, in a potentiometric detection system, the change in the pH can be measured, or as it says here, the change in the sodium ion or the change in the potassium ions can be measured. Okay, so therefore the concentration of the analyte can also be detected through this enzymatic reaction. So that is what the job of an enzyme sensor is. So such sensors you are going to call as enzyme based sensors. They are not microbe based. So now let me move to the next slide, where we will talk about, so what we looked at earlier were electrochemical transducers. But in biological systems, we also make use of these optical transducers, okay, where lights are going to be produced, and these are going to be measured in a device. How much of light is going to be produced will directly be a reflection of some kind of a microbial activity which occurs in response to the presence of a certain pollutant. Okay, so those are called as optical transducers. Now let me begin with as to how can you use these optical measurement system. How can microbes be utilized for that? So here is a picture of a bacterium. Say it is a Vibrio harvey, okay? And as you see that these bacteria are luminescent. They are able to produce luminescence, which can be measured in a luminometer, okay? However, the production of this or inhibition of this luminescence can be employed in the optical transducer system. Okay, so let me just read out that many bacteria, like you read here, Vibrio fischeri or Photobacterium or Vibrio harvey or Pseudomonas fluorescens, as the name suggests, the name of this bacterium, the bacterial genus is Pseudomonas, but the species name is fluorescens. Why? Because it fluoresces. And I will give you what is the molecular basis of fluorescence. And number two, how this property in a bacterium can be employed. It can be, applied to develop a biosensor. Okay. So the popularity of this bacterial assay is based on the fact that bacteria are an integral part of the ecosystem, and the bacterial assays are very quick and they are very simple to conduct. So in other words, the idea is that can you have, like, for example, these particular bacteria, and you just suspend them in a sample of wastewater, and suddenly if it starts to fluoresce, that could indicate that there is a contaminant which is present, there is a pollutant which is present in that wastewater sample. So that is the entire fundamental behind this. Okay, so let us say you want to know if a water body is contaminated with mercury. So what I'm going to do, I'm going to use this Vibrio harvey as a biosensor. I put a suspension of these cells into this wastewater, and suddenly I find that this bacteria is able to fluoresce. Okay, and that would tell me, and how much fluorescence is exhibited could be a measure of how much of mercury contamination is in that wastewater. But if it does not fluoresce, that means there is no contamination of mercury in the wastewater. And you could do it with for heavy metals. You could also do it for aromatic compounds like naphthalene or salicylic acid or aromatic hydrocarbons, etc., where the cells or the luminescence of the cell is going to be used as a measurable parameter, which will tell you the presence of that pollutant. So is that concept clear to everybody?
Yes, ma'am.
All right. So now the growing, and the question is that you may wonder, how can you just oversimplify? What makes you sure that if a human being if a bacterium is going to respond to the presence of this, inhibition of fluorescence means that there is a compromise on the biochemical activity of a bacterium. How can you be sure that how the bacterium is affected? A human being will also be affected in the same way. Well, the question is that it has been found that a substance, what is written in red, I'm just going to read it out to you. A substance which is toxic for an organism often demonstrates similar toxic effects on other organisms also. Okay, so something which is going to be inhibitory to a bacterium, all right, can also be inhibitory to, for example, fish, or it could be very much inhibitory to, that is, if you just adjust everything to milligram per kilogram body weight. That is something which you will have to take into account. Now, bacterial cell mass is very, very small. So you have to make sure that you are looking at the same amount which is subjected to per milligram or per gram of body weight. If you adjust that, then there is no reason for us to believe that what appears to be inhibitory to the bacterium will not be inhibitory to a fish or to a mammalian system. Okay, so in other words, the luminescence inhibition in a bacterium can effectively indicate the toxic effects on higher organisms also. That is what the rationale is of this. Now, first of all, I will just explain to you as to what is the biochemical basis of luminescence in these bacteria, and then we will see how we can exploit this luminescence. So if this slide is visible in this mode, I will not go to the full PPT mode. So let me see if you are able to see. So are you able to see the slide well?
Yes, ma'am.
All right. So now this bioluminescence phenomena, which we see in the bacteria, okay, is something which happens because of the following events. All right. And this is something which only occurs, for example, it is a function of the cell density. Under, like it says here, under low cell density, when in an aqueous solution, Vibrio fischeri is present in very low numbers, okay, then what happens? That they have an operon which is called as a lux operon. Okay, lux is actually for luminescence function. All right, and since it is an operon, that means in the operon there are several genes, I will show you in the next slide, which are a part of the lux operon. Just like in the lac operon, you will remember you had the Z, you had the Y, and you had the A genes. Similarly, in the lux operon also, there are several genes which are present in this yellow line here. Okay, and then there is a regulatory gene which is called as there is lux I. It's not a regulatory gene, but it is another gene which is a part of this cluster, and the product of this gene is responsible for synthesis of a molecule which is called as homoserine lactone, which is shown as these pentagons here, which are red colored. Okay, and then upstream of that is what is called as a lux box, which is just a DNA sequence. It doesn't code for any protein. Upstream of that is another DNA segment, which is called as the lux R, and it codes for the lux regulatory protein. Okay, just like in the lac operon, you had the lac I, which was coding for a repressor protein. Okay, so in your mind, you have to bring back the concept of operon and what is a regulatory protein. Okay, now going back to what happens under low cell density. Okay, the production of lux R, it is constitutive. It is constantly made in the cell at very low concentration. However, under low cell density, the production of homoserine lactone is very, very low. So the molecules of homoserine lactone, they diffuse out of the cell. They can also come back inside the cell, but the concentration is very low. Under these conditions, the lux operon is not activated. So therefore, there is no luminescence which is occurring in this picture. But under high cell density, what happens is that when there are too many cells, then the concentration of this homoserine lactone, for example, builds up to a very high level in the extracellular medium. Okay, since many of them they can freely enter inside the cell, the concentration of the homoserine lactone also goes up inside the cell. Under these conditions, they can bind to the lux R. There are too many molecules here within the cell. The lux R is able to bind to them. The lux R, now the repressor protein, gets activated when it binds to this homoserine lactone. The activated repressor will bind to lux box. Okay, so these are just conformational changes which are occurring. So the DNA sequence could not bind to this. I mean, that should not come as a surprise to you, because remember how specific restriction enzymes, they will act only on a defined sequence of DNA. So protein DNA interactions are highly specific. Okay, the minute changes in the conformation can trigger a lot of activity. So when this homoserine lactone binds to lux R, it changes its conformation. Under that stage, it can now bind to this DNA sequence. So this gets activated. All right, and the transcription of the gene begins here. That means in the lux box, RNA polymerase is going to bind, transcription is going to occur, and all the genes of the lux operon are going to be transcribed. Proteins are going to be made as a consequence of the activity of the various enzymes. Now luminescence occurs. Okay, so is this slide clear to everybody?
Yes, ma'am.
Okay, so luminescence, you may ask, what does it do for the cells? Okay, so the phenomena of bioluminescence allows the cells to gauge their population. It is called as quorum sensing. So in a solution or in a large water body, the bacteria are able to gauge what is their density. How many of them are really present? They don't have any feelers, they don't have any antennae, etc. But what they do have is that the luminescence, this phenomena of luminescence can be perceived the light, the photons which are going to hit the cell surface. Now they are able to gauge that the cell membranes are sensitive to photons. They are able to gauge that now luminescence is being produced. That means there are a lot of us in this medium. So basically luminescence has evolved as a mechanism for quorum sensing. So what do the bacteria use it for? The bacteria use it for causing virulence. If there are too many of them, they are capable of causing infection in the host. Okay, horizontal gene transfer, the genes which are involved in gene transfer, that is conjugation activities, are because if two bacteria are lying 2 meters away, they don't want to synthesize enzymes which are responsible for conjugation event. When the density is large enough for them to conjugate, only then the enzymes which are responsible for conjugation, such as the filament formation or the pili formation, only then they are going to be induced when the cell density is very high, or for example, the biofilm formation. Okay, because then they can form a biofilm, so on and so forth, or competence, that is the ability to take up DNA. So all the genes which are responsible for competence, for giving competence to the bacterium, biofilm formation, conjugation events, or causing virulence, etc., they are all dependent on how many bacteria are there. So this is a way for bacteria to gauge their population and to control their behavior. So we say quorum sensing is a key behavior coordination mechanism in many microbes. Okay, so as a principle, this is what you first need to understand, that of course I want to use this bioluminescence in the form of a biosensor. But before that, I should also understand what causes bioluminescence and what are the biochemical events which lead to bioluminescence. So in the next slide is the chemistry of the bioluminescence. Okay, so I hope all of you can see the slide.
Yes, ma'am.
So if you have any questions, because it is a fascinating topic today, very much like a circuit. So people are designing their circuits, people are designing microbial activity by way of bringing different genes, putting plasmids in different kind of configurations in order to build circuits which can now respond to the presence of a certain molecule in the environment. So let's look at, so this is what you have already seen. And as a result of luminescence, this is what the bacteria is going to look like on a plate. So you can actually see these luminescent bacteria are going to look like this. So the chemistry behind the reaction. I talked about the Lux operon. Now let us see what are the genes which are in the Lux operon. All of these genes are put down here. These are Lux C, Lux D, Lux A, Lux B, Lux E, and FRP. So in other words, in this Lux operon, there are 6 genes which are located there. All right. And what is the basic reaction which is being catalyzed during the production of light is shown to you here. That is, so first we have this is an flavin mononucleotide which is in the reduced form. So the reaction which is occurring during the light production is FMNH2 combines in the presence of molecular oxygen with an aldehyde, with a long chain fatty aldehyde inside the cell, and then bacterial luciferase, which is actually coded for by Lux A and Lux B. So 2 polypeptides are coded for. These 2 polypeptides will come together to form a heterodimeric enzyme. And this bacterial luciferase then will convert, will carry out a reaction in which FMNH2 is oxidized to FMN. Aldehyde is now, you have, reduced it to acid, and water is formed. Okay, and as a consequence of this reaction, light is emitted. Okay, so this is the actual chemistry behind the reaction. So there is a lot of energy, a lot of the photons which are released in the reaction, like it says here. So if you want to look at the same reaction, we have like aldehyde, which is the long chain fatty aldehyde you see, plus FMNH2, which is analogous to this FMNH2 plus oxygen. In the presence of a bacterial luciferase, you have the long chain fatty acid which is formed. You have FMN, so this is now oxidized to FMN. Oxygen is reduced to water, and photons are emitted of a certain wavelength, 490 nanometers. Okay, so that is how the light is emitted. Now, what is the function of the other genes which you see in the lux operon? Okay, so in order for this fatty aldehyde, because you see every time in the reaction there are fatty acids which are formed. Okay, so in order for the cells to have a continuous supply of fatty aldehyde, the other enzyme which is responsible for converting the long chain fatty acid into long chain fatty aldehyde, for the luminescence to continue, you need to recycle this fatty acid which is formed into aldehyde. That is carried out by the gene product. So this is a protein which you will see here: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it is a protein which has 12 different subunits. It's a multimeric protein. Okay, one polypeptide, four of which are present in this polypeptide is coded for by the gene LUXC. So no, mustard, gives you this right here. Okay, so LUXT, it is shown in the mustard color. That means it codes for this particular polypeptide. This is shown in the green color, LUXC. LUXC codes for this particular polypeptide. Okay, and similarly you have the light blue one which is coded for by the lux E. So the products of these three genes, so you have four copies of this coming together, four copies of this, and four copies of this which are coming together. Okay, in order to make a multimeric enzyme which is called as fatty acid reductase complex, which changes the fatty acid into an aldehyde. So this is the function of these three genes. Okay. Similarly, in order for luminescence to occur continuously, FMNH2 needs to be now constantly regenerated. So this reaction of converting FMN into FMNH2 is carried out by the enzyme flavin reductase P here. And this is coded for by the FRP enzyme. It's a monomeric enzyme, and so this reaction, so that is how all of these enzymes, for example, these three enzyme clusters are able to produce luciferin. And how do they get activated is only because the lux operon is getting activated by virtue of what events are occurring here. So bioluminescence is produced. All right, so is this clear to everybody, this slide?
Yes, ma'am.
So now, you have two options. As we use this luminescence phenomena in the environmental biotechnology, there are two things you can do. In the first one, what you could do is that if luminescence stops, that means the biochemical activity of the bacterium has stopped. Why? Because there could be a pollutant which is present in the environment. So bioluminescence directly can serve as a measure of the pollutant concentration. So that is what is written in the first part. Luminescence in these bacteria, the Vibrio fischeri, can serve as a measure of the physiological activity of a bacterium. So if there is a pollutant that inhibits the bacterial luminescence, all right, is indicating that physiologically it is a pollutant and it is causing some harm to the life form. So that is one way of using luminescence as a biosensor. Okay, the other way is that luminescence can also serve as a reporting element for the presence of a pollutant. In other words, if the sensor element of luminescence gets activated now, not by the presence of homoserine lactone, but if it gets activated by the presence of a specific pollutant, such as a metal ion or an aromatic hydrocarbon, so is the first point clear to everybody?
Yes,
Yes,
ma'am.
ma'am.
First point is clear.
And what about the second one? Okay, because you see, if you look at, for example, this particular slide, all right, now in this slide, there is an element here. You see the Lux box, or let me go to this bigger version of it. There is a Lux box here. So what it is doing essentially is that it is sensing the presence of a molecule homoserine lactone and activated lux R. Now what about if in front of the lux operon, I put an element here which senses the presence of mercury ion, which could combine with another repressor molecule, and the sensor element is very, very different now. So rather than sensing the presence of lux R and homoserine lactone, what if this sensing element, the DNA sequence here, okay, senses the presence of mercury? Then as soon as mercury binds to this site, the lux operon will get activated, and these cells are going to there will be bioluminescence which is produced. So in other words, now the lux operon can be used to sense the presence of a metal ion in the environment. So is that concept clear to everybody? Do you see how it can now be used as a biosensor, but in the other direction? So, everything to the development for example, of a biosensor like for based on bioluminescence is actually based on this concept. So here you need to understand what is the function of this DNA sequence and how it doesn't matter, any DNA sequence can now be just ligated in front of the lux operon. Okay, because the idea here is that it needs to open up, the DNA sequence needs to open up for an RNA polymerase to bind here, so that now transcription of the lux operon can occur inside the cell. And this you could do even in an E. coli. It doesn't have to be in the original Vibrio fischeri. In E. coli, what if you clone the genes on a plasmid of the lux operon, and now you have some other DNA sequence right here, which can open up, which can bind to RNA polymerase. But this particular DNA segment is now going to be sensitive to something which is other than homoserine lactone. So is that point clear to everybody? Okay, so students, you have to give a response that
Yes, ma'am,
have
it
you
is clear.
understood? So therefore, so everything which we are going to talk about now will be really based on this concept. So keep this in mind. So now, so these are the two ways that the luminescence can be used now to develop a biosensor. Let me give you an example of the first category, where Vibrio fischeri, for example, or another species, which is Vibrio harvey, can now be used as a sensor because there is inhibition of fluorescence, which tells you that there is some kind of a pollutant which is present, which is inhibiting the normal physiological activity of the cell. So therefore it is a pollutant because it causes an inhibition in some kind of a biochemistry. So Vibrio harvey is not able to give as much, like unlike in the in the control sample, where when you incubate your cells in simple water, there is a certain level of fluorescence which you are going to get. But now if in your test sample you are going to add a pollutant and there is an inhibition of that luminescence, that means the water which you have, the things which you have added, or the environment which is surrounding the cells now contains a pollutant. Okay, so what is measured is now inhibition of bioluminescence, and it can be calculated by this particular formula, where you have the fluorescence which is of a test sample which is measured. It could be a number like maybe 40 or 50, which is after incubating it for a period of time which is described as T. So let us say you incubate your Vibrio harvest system for a period of 40 minutes as per the standard test. And now you see what is the intensity of the fluorescence. And you measure what was the intensity at time 0. So as soon as you added your cells, you note down what is at time 0 and what is at time T. Kf here is the correction factor, which is correction factor means that, if you were to take the control samples, that is, if the same cells are suspended in water at time 0, do they give any fluorescence? Or at time T, if they are still kept in the water, do they give any fluorescence? So that is called as a correction factor here. So I hope you understand, what are all of these terms, what do they stand for? So if you measure all of these parameters, you will be able to measure what is the percent inhibition. Okay, and the percent inhibition, for example, is also represented by EC50 value, which is the effective concentration or the inhibitive concentration. So in other words, you want to measure what is the concentration of that pollutant at which 50% of the luminescence is inhibited compared to what you see in the control cultures. So the concentration of the toxicant in milligram per liter, which causes a 50% reduction in light after exposure for T minutes, is designated as the EC50 value. Okay, and this luminescence emitted can also be measured by luminometers, which are typically provided with the commercial test kits such as Microtox, because I had told you that these are the standard kits which are available, and luminometer is also a part of that standard test kit. So the data which is shown on the right here tells you what is the sensitivity of the bioluminescence when it is compared to many other standard tests which are already available in the market. How does it really fare? If you take, for example, Vibrio Harvey, as an indicator of the level of the pollutant. So here is the data which is given for cadmium. So the pollutant here is either, cadmium, heavy metal, or chromium, or copper, or zinc. And the 5 histograms which are shown here are 5 different tests which are being carried out. The one in the black is the nitrification inhibition. So it's a different group of cells which is capable of carrying out nitrification. Okay, so if you so it's a different bunch of cells altogether. So if you take those cells and you incubate it with cadmium, for example, then the EC50 value or the IC50 value which comes out in this test, this is the log scale. Okay, so the value is about 20 now or 30. So at 30 milligram per liter of cadmium, in this test, you have 50% inhibition of luminescence. Now, the second bar which is shown here is a test which is a respirometry based test. That is, the cells are suspended in a medium and you measure what is the amount of oxygen it takes up. So if there is a pollutant, like if there is too much of cadmium in there, then it is going to act as a poison, as a result of which the level of oxygen uptake is going to be lowered. So you see what is the level at which there is 50% inhibition of respiration. That is what is measured in the respirometry test. So if you carry out, the cadmium, for example, at a given, different concentrations you carry out and you find out what is the IC50 value. So through respirometry, the value, is the IC50 value is approximately, let's say 60. Okay. The third test which you see there is the ATP luminescence, which is carried out with another bacterium. The Vibrio fishery data is shown here, and enzyme inhibition. So you take some enzyme and you add this pollutant to it and you see what is the enzyme activity. So these are, in other words, 5 different tests as are explained here. The numbers which are given here, for example, are 3 tests were in triplicate. Then this is carried out, in 2 tests. This is in 2. This is 22 times it has been carried out, or 6 times it has been carried out. So the numbers which are in the parentheses, they actually refer to the number of times that the test has been carried out. So what you see here, the data indicates that this one is the Vibrio fishery. It's extremely sensitive. That is, the lower the EC50 value, that means the more sensitive the test is. Even at lower concentration, about 8 milligram per liter of this, it is able to detect even the lower values of this are able to cause 50% inhibition. So this test is more sensitive compared to so many other tests which you have carried out. That's what the data indicates. So is that point clear to everybody as to what this data is trying to tell you?
Yes, ma'am. We are going through the slides.
All right, so you are able to understand, because I don't know, sometimes my net is bad, so I don't want that I should be speaking and the connection is not there. So in this particular picture, again, you are looking at the copper as a pollutant. Here you are looking, so all of these are basically heavy metals. So you are just testing, because remember I told you, why do you worry about heavy metals? They are also pollutants, especially from the painting industry. All these heavy metals are released into the environment, so they are also pollutants. So that is what is represented here. So in all of these tests, what you see is that the Vibrio fishery test here is extremely sensitive. So IC50 can be achieved even at a much lower concentration. So that shows that, you are able to pick up extremely small quantities of these pollutants, which gives you some kind of a phenotype, some kind of an effect on a biological system. Then similarly, in the lower figure here, they have used exactly the same tests, but for detecting the presence of organic pollutants like 3,5-dichlorophenol, toluene, LAS are the linear alkyl benzene sulfonate compounds, which are a byproduct of the petroleum industry. Or then you have the mixtures of all of these, and again you have a mixture, so mixture 2 has a different composition, mixture 1 has a different composition of these three. So in all of these, the idea here is to let you know that the Vibrio fishery has emerged out as a very successful test. So all you are doing is that you have the cell suspension Vibrio harvey, and as the cells are going to multiply in a control culture, in a normal healthy water environment or in a medium, they will multiply. And when the cell density builds up to a certain level, due to quorum sensing, they are going to give you fluorescence. So after a certain time period, you are going to detect a certain amount of this luminescence, which can be measured in a luminometer. So let's say you have 20,000 units for a control sample. But now in the same suspension, you are going to add a pollutant, like you will add cadmium or chromium, incubate it for 40 minutes, because it has an adverse effect on the cell physiology. So the luminescence is not going to be as much. The cell density which you will achieve will not be as much as in the control sample. So that decrease in the luminescence serves to tell us that there is the presence of a pollutant. And how much, eventually you are going to quantify everything on the basis of IC50. Because all data is not like 40% inhibition, 50% inhibition. No, all the data is reported in terms of IC50 values. So that is how across the different laboratories you are able to compare the data. So now, if this is clear to everybody, let's go to the next slide. Okay, so now I'm going to give you the example of the second use, that how the Lux operon can be used as a biosensor, for detecting the presence of all of these pollutants, but by molecular biology way. Not the whole cells are going to be using, but you are going to detect the presence of these through a natural system, by intervention, not using a natural system, but through a recombinant way. And we will see as to what is the advantage of that recombinant way. Okay, so in order to understand that Lux operon you have already understood, but now what I'm going to give you an example is that how Lux operon can be used to sense the presence of naphthalene in the environment. Okay, so now in this slide, in the upper slide is represented as to how in many bacteria like Pseudomonas, for example, is able to break down naphthalene, and the metabolites which are generated from naphthalene can be now fed into TCA cycle. So that is how naphthalene can be used as a source of carbon. Okay, so a bacterium like Pseudomonas, so this is the picture of naphthalene here, all right, and the green arrow represents many enzymatic steps which are carried out in order to break down naphthalene into salicylic acid. And all these enzymes are coded for by genes which are a part of what is called as an upper operon. And this operon contains the genes which are shown as NAHABFCED. What reactions do they catalyze are given here. Okay, so when naphthalene is degraded into salicylic acid by a bacterium like Pseudomonas, for example, so what it does here is the naphthalene right here. So the product of the gene NAHAB right here, NAHAB, the gene product is able to convert naphthalene into this hydrolysis. So it is a dioxygenase. So you see 2 atoms of oxygen are added here. At this position, at this position. Okay, so I told you earlier also about monooxygenases, that all aromatic compounds, when they are broken down by the bacteria, the action usually starts by adding a single oxygen by enzyme like monooxygenase, or 2 atoms of oxygen are added by dioxygenases. Okay, so now this particular compound is acted upon by the product of the NAHC gene, which you see here, NAHC gene, which converts this into this, then NAHD gene, which is right here, okay, converts this into this, and NAHE, what you see here, E for example, will convert it into this, and NAHF, which you see here, will convert this into salicylic acid. Okay, so now it should make sense to you as to how in a bacterium like Pseudomonas, naphthalene is converted into salicylic acid. So many enzymatic reactions are carried out. All of these are coded for by genes which are a part of this particular operon. It's also called as the upper operon. And then salicylic acid is converted into a TCA cycle intermediate. Okay, like some maybe fatty acid like acetyl coenzyme A, which can enter into the TCA cycle, or it can be converted into something else, an acid. You have alpha ketoglutaric acid, you have citric acid, you have the malic acid, malate, which is a part of the TCA cycle. So any one of these intermediates, if they are formed from salicylic acid, can enter into TCA cycle, and they can be completely mineralized into carbon dioxide and water. So for converting salicylic acid into a TCA cycle intermediate, these are the different gene products. NAHG, which is a part of this. Okay. Similarly, NAHH is written here, NAHI is written here, so on and so forth. So all of these enzymes are going to act sequentially on this in order to convert it either into an aldehyde or into a fatty acid, which can now, this is like, alpha ketoglutaric acid, or something which can now enter directly into TCA cycle. Okay, and then right here you have a regulatory protein, because activation of these operon requires the participation of maybe naphthalene and a regulatory protein which can bind to these promoters and activate these operons. Because remember that these genes are not going to be transcribed unless naphthalene is in the environment. Just like when glucose is added into the culture or lactose is added into the culture, only then the lac operon gets activated. So is that point clear to everybody?
Yes, ma'am, it is clear.
Okay, so now what I'm going to show you is that how a sensor can be constructed based on your knowledge of how lux operon, entirely different physiology, naphthalene, which is from another organism, it is from Pseudomonas. Lux operon was in Vibrio. We're not talking about Pseudomonas fluorescens, which has this, but we have some other strain of Pseudomonas, which is capable of breaking down naphthalene, but it doesn't make luminescence at all. So these two diverse systems are going to be brought together in an organism, okay, which will be capable of metabolizing naphthalene, and at the same time, they will also couple it to luminescence phenomena. So let's see how such a construct can be made. So in this particular picture, pay attention that this particular plasmid which you see is taken from a bacterial cell, E. coli. Okay, so the plasmid UCD623 is present in a bacterial cell, which is E. coli HP101 cell, which contains this plasmid. The plasmid contains this gene. Okay, and what does it contain? It contains the lux operon. On this side, for example, is the plasmid which comes from a Pseudomonas fluorescens strain, okay, and so this is a drug resistant strain, and this particular plasmid is taken. You see here. So this particular plasmid now contains the genes for upper pathway, which you saw. Again, I will remind you, upper pathway means that this particular operon, and it also contains a lower pathway. So these two clusters which you see are nothing but this and this on this two plasmid. These two cells are allowed to mate with each other. Okay, and how is it done? On a petri dish, for example, imagine that you have a petri dish which has Lb plus agar. So horizontally, you draw a strip of cells which is actually the E. coli HP101 cells. And no, vertically you draw the cells, and horizontally what you are going to draw is with a toothpick or something, you draw out a cell which is actually the Pseudomonas fluorescens, which contains this particular plasmid. So, just imagine that you now have a plus sign. So you have, for example, cells which are coming this way, the cells which are coming this way. It is only in this particular area in the center that these cells are allowed to undergo mating with each other. And recombinant cells are now picked up on a medium which contains both, it contains both tetracycline as well as rifampicin. Earlier, only like, the E. coli 101 that contains, for example, this one of the genes which is tetracycline, and this brings in the rifampicin gene. So on the medium or in between, you are going to select for those recombinant cells which are capable of growing on both rifampicin and tetracycline. So, you're going to select now for a recombinant cell, not for any one of the parental cells on such a medium. Is that clear to everybody?
Yes, ma'am.
Okay, so that particular recombinant cell was taken up, and plasmids were isolated because they were luminescent. Okay, so these cells were now luminescent, and eventually, the plasmid was determined as to what happened. So let me now just show you the experimental results which are obtained. Okay. So this particular recombinant strain, which combines the property of having a lux operon as well as sensitivity to naphthalene, was called as the strain HK44, and it contained the plasmid PUT-K21. Okay, so these results were obtained in a continuous culture. So what is done is that the recombinant cells which you have got out of mating of E. coli HP101, which contains the lux operon, and you have now Pseudomonas fluorescens, which is capable of metabolizing the naphthalene. Okay, so the recombinant cell which you have is now cultivated in a bioreactor on a non-naphthalene carbon source, and it is allowed to come to a steady state. So in a chemostat, for example, the cells are allowed to grow until, let us say, they are now fed on glucose. So the glucose is fed and the cells achieve a steady state. At that particular stage, gradually what is done is that you see in the circle here, for example, is the bioluminescence, and in the triangle what you see. So at this particular stage, naphthalene is introduced into this chemostat, where the cells have been allowed to come to a chemostat, but on a non-naphthalene substrate. So as soon as naphthalene is added into this environment, okay, there is luminescence which is recorded now. So the solid circle here represents, for example, the bioluminescence which is shown by a recombinant culture only in response to the presence of naphthalene in the medium. Okay, so you have, for example, luminescence, and what you have here, for example, is the rate of naphthalene degradation rate. So if you see, what are the units? So you have about 9. So under these conditions, there is naphthalene degradation rate of 9 milligram per hour in this particular system. Okay, so as long as naphthalene is in the medium, the bioluminescence occurs. Initially, it increases and then it comes to a steady state. So the degradation rate is so much, and the bioluminescence is now produced at this particular rate. Okay, so in other words, what is so unique about this system is that the recombinant cell, it is capable of breaking down naphthalene, but it was not a bioluminescent strain. But now it is able to use naphthalene as a source of carbon, and it is able to build up enough cell density to such a level that now the bioluminescence phenomena is exhibited. And it is only in the presence of naphthalene that this phenomena occurs. So,
Ma'am, I had a question in this slide that the bioluminescence is a measurement of the cell density, and the naphthalene
right
is
right now, the bioluminescence in this particular construct, the bioluminescence is not a measure of the cell density. It is a measure of naphthalene degradation in the system.
but ma'am, the genes are different for naphthalene and the bioluminescence. So how can the naphthalene degradation can affect the bioluminescence?
Exactly. So I will come to that now. Before I return to that, let me give you another. The result from this particular paper is a dynamic response to 4 hour perturbation. Okay. Uh, See, because the activation of that Lux box operon, okay, which occurs naturally in response to homoserine lactone, is a density dependent phenomena. But it doesn't have to be like that. It can be that sensory element can be something else also there. All that is the end product which is required is the unwinding of that particular segment of the DNA molecule so that RNA polymerase can bind to that region which is just preceding the Lux operon. That anything could do. All right. So what I will do, let me just explain this figure and then I will come back to explain as to what happened in those recombinant cells. So the cells are again cultivated in a chemostat and they are allowed to come to a steady state on a non-naphthalene substrate. And you are doing it in a steady state. So the cell density, etc., are all kept constant. The only thing which is going to change now is that there is a new substrate which is being brought in. Okay. So So the straight lines, they refer to naphthalene feed concentration. Okay. So at this particular site, okay, so the cells are in a chemostat and you allow the naphthalene, the feed naphthalene concentration milligram per liter. So, there is a steady state concentration like 18 milligram per liter where you are doing this, and there is a steady concentration of bioluminescence which is occurring. So in this particular ride, at this stage, what you are doing is that you are increasing the concentration of naphthalene, the feed naphthalene, from 18 gram per liter to about 30 gram per liter. You're not changing anything else. As a result of which, there is an increase in the bioluminescence. And triangle, what does it represent? Is the reactor liquid naphthalene concentration. As the feed increases, you have a feed rate, so the feed increases, but then you just remove a sample and you measure the luminescence of the cells, and you see obviously this is going to follow an increase in the feed concentration. So you see that there is an increase. It is kept constant. Now the feed concentration is kept constant for a certain period of time at 30 milligram per liter, and under these conditions the fluorescence continues to increase. But now you reduce the feed to zero. At this time point, suddenly the feed is cut off. It's switched off and only some medium is going in there because it's a chemostat. Except for naphthalene, everything else is going in there. Okay, and what you find is that the cells are exactly the same, and now there is no luminescence which is produced. So when naphthalene is completely removed from the medium, the bioluminescence also decreases, and that is why we say that it is this system is responding to the presence of naphthalene. So it's a naphthalene biosensor. These cells are naphthalene biosensors. They don't respond, they don't change in response to cell concentration, but the luminescence changes in response to naphthalene concentration. So is that clear? So similarly, again, they have done it over this period. So naphthalene is complete. So at this point of time, again, it is changed to 30 milligram per liter in the feed. In the reactor, the concentration increases, the bioluminescence increases until the feed is completely withdrawn. At that stage, again, the naphthalene concentration in the feed, of course, goes low as shown by triangles, and also the bioluminescence decreases. And that's why we call it as a naphthalene biosensor. So is that clear? And now let us see what exactly happened during the recombination event. Okay, so in the recombinant cells, you have the plasmid, which was later on isolated, sequencing was done, and whatever methods were available, it's a relatively old paper. So whatever methods were available, the structure of this particular plasmid, which is present in the recombinant cell, was now determined. And it was something like this, that you remember that these are the two naphthalene, the upper pathway and the lower pathway. So this particular gene cluster, which you see here from E. coli HP101, seems to have entered at this particular location, which is just upstream of NAHG gene. Because these are the genes of the upper pathway. NAGH is a part of the lower operon, and this is the regulatory protein. So the Lux operon seems to have entered right here. Okay, so this is where the Lux operon was located, and TCR is because there is a tetracycline resistance gene also at that location, and then there are transposon based genes here. Okay, so because of insertion of this, okay, and this is, they have left the promoter of this is left intact. Okay, so what happens is that just downstream of the promoter, which is this particular promoter, which is the salicylic acid promoter, you have inserted actually a Lux operon. So as soon as, this promoter is very sensitive to the presence of the repressor protein plus salicylic acid. Okay, so there are constitutive levels which are always produced in Pseudomonas, which are able to convert a little bit of naphthalene into salicylic acid. Okay, that salicylic acid binds to this, it activates it. But since you have now a Lux operon, so it is the Lux operon which is getting activated. Apart from these genes, this Lux operon is getting activated. And that is why every time there is naphthalene metabolism to salicylic acid, then the Lux, or there is naphthalene which is availability there, all right, then the system is going to respond by producing fluorescence because the Lux operon gets activated. However, the activation of the Lux operon is actually dependent on what happens to the DNA sequence here. That is, the DNA, double-stranded DNA has to get unwound and RNA polymerase has to bind there in order for transcription to occur. It has nothing to do with the Lux box. That could be any DNA sequence. So is that clear to everybody as to how it happens?
Yes, got it.
So now let me very quickly give you some other example. So you see, so in this kind, what you are doing now is that this Vibrio fischeri system or the bioluminescence system. So, Lux operon can be cloned in an E. coli cell. You can develop E. coli as a biosensing agent as long as the Lux operon is there, and the promoters which are upstream of that, they respond to specific elements which are derived from the original promoters which are present in the nature. So here I'm giving you an example of a mercury sensing element, because there are several bacteria which are able to break down mercury. By breakdown, that means they are able to complex mercury and convert it into very inactive heavy metal. They are not poisoned by mercury. And that is done by the presence of several enzymes, okay, which are like, one operon is called as the mercury TPCAD, and this is the right, this is the arrow which indicates the transcription of the mercury operon, and this is because any gene before it can be transcribed has to have a -10 element and the -35 element. So these two, you see, the promoter which are right here, they are actually corresponding to this particular gene. At the same time, there is a mercury regulatory element, a regulatory protein. So just like you looked at the Lux regulatory protein or the LAC repressor protein, etc. So there is MER-R, which makes the MER-R protein, which actually binds with mercury. And let me tell you as to what happens. Okay, so for this MER-R, this is the -10 and -35. So these are divergent promoters. The direction of transcription of this operon occurs in this way, and the direction of transcription occurs in this other way. And that is what the arrows are indicating here. on this cluster. So the way the biological system from which this is taken acts is that MER-R codes for a repressor protein. Okay, and this repressor protein, for example, you see in the absence of mercury, for example, okay, so there is a repression. That means when there is no mercury which is present in the environment, the repressor proteins, they form a complex in such a manner that they are not able to bind to this, they bind to DNA in this particular manner, so that this particular DNA segment doesn't, the RNA polymerase, is not able to bind to a complex. The DNA is no longer bound to the -10 site here. Okay, but what happens in the presence of mercury? When this biological system, when this microorganism, when mercury gets inside, it is able to bind to the repressor protein in this altered conformation. When it binds to this site, it brings the -35 and -10 in position on RNA polymerase. So now transcription can begin. And all the genes which are downstream, that is the products of MER-T, MER-P, MER-C, MER-A and MER-D. The products of these genes are going to be made, and those enzymes will be able to now act on the incoming mercury and make it non-functional, because they secrete certain kind of chelators which is able to bind to mercury so that there is no free mercury. Otherwise, mercury can act on a lot of enzymes. They have sulfur. So may all proteins have the cysteines and other things. So mercury acts as a poison by acting to these sulfur containing amino acids, and it can poison enzyme activity. So what they do is that they secrete a lot of these enzymes, which could be sequesters, which could be, altering the mercury capacity so that it is no longer able to bind to the protein. So this way they inactivate mercury, and they are said to be mercury resistant bacteria. So is that clear? So RNA-P stands for RNA polymerase. So is this clear to everybody?
Yes, ma'am. Yes, ma'am. It is clear.
Okay. So now, you can imagine that if the gene for MER-R, or if this particular, if this site, let's say this particular site, okay, and the gene for MER-R, only this particular site, if it is put next to a Lux operon, then the Lux operon will be activated when the repressor protein binds to this. You see, it doesn't care. All that it cares is that the DNA has to be unwound so that transcription can begin. So what you put upstream could be very, very specific of the Lux operon. It could be a DNA sequence which you derive from the original mercury operon, from the naphthalene operon, from aromatic hydrocarbon operon, or from cadmium sensing operon, or whatever. As long as you put those sensory elements here, which can bind to their own repressor proteins and that metal ion, it is able to cause unwinding of the DNA so that transcription of the Lux can occur. Okay, that Lux operon therefore can serve now as a biosensor for any one of these that you want to choose. It could be a hydrocarbon, it could be a metal ion, or whatever. So a biosensor can be developed on the basis of that. So I'll just stop here now, and this will also be like a part of your minor exam. So if you have any questions, you can post them, or if you find time, some of you, we can again come back on the Microsoft Teams for half an hour for a doubt clearing session. So please go through the lectures. The reading material for this also I will upload. So just try to understand the basic working of the Lux operon and how the different units, they can be intermixed. They can be, combined artificially, because today you can ligate DNA fragments, etc. So this allows you to build up your own biosensors.
Ma'am, can we have a doubt clearing session on 13th? Because ma'am, I'm going through the readings, so I haven't not completed
Certainly,
yet.
certainly. So I think just circulate a mail on the chat that, on 13th and Sunday you decide a time because I would prefer maybe like, 11 o'clock in the morning or something like that. So if all of you want to assemble on Sunday morning around 11, we can just have a doubt clearing session.
Yes, ma'am. Sure. Thank you, ma'am.
Okay, so please take the consensus from other students also, and then we can come back. Okay, fine. And are there any questions related to how the exam is going to be conducted?
Yes, ma'am. Like for the online students, what will be the pattern of the exam?
Yeah, pattern of the exam I have already given you. That is the kind of questions which are going to be there will be short questions and very specific to the point. And as I said, the question paper is going to be uploaded on the Teams. I will also send you a copy of the question paper. Okay, and obviously, please keep all your WhatsApp and all your telephone devices and everything, keep the video on. And then once you have written, let's say over 4 pages or over 3 pages, all your answers are written, then you take a picture of that time with your mobile camera, put it into a single PDF document and send it to me by email. Email address I have already given you. Or you can WhatsApp that PDF document to me on my mobile.
Ma'am.
Yeah.
Ma'am, the end time is given as 10 a.m. So we will start at what time, ma'am?
I think I had written we will start at 8:45. मैंने कहा था
Yes, ma'am. Yes, ma'am.
8:45 the exam
Yes,
will
ma'am.
begin, and you
Yes,
will
ma'am.
get
Understood.
15:20. Yeah, and the next exam for you is not till maybe 10:30 or something if somebody has a back-to-back exam. So you will have enough time to finish this and move to the next exam. Is that clear?
Yes, ma'am. It is clear.
Okay. Or anybody else, people don't come forward to talk. Everybody is on mute here. I don't know what's happening. And, suddenly you are flooded with so much of reading material. So if you have any other questions, just please post it on the Teams.